Everybody. This afternoon we are going to discuss about microbiology, and let's go right into the introduction. Definition: Microbiology studies microorganisms which include bacteria, viruses, fungi, and parasites. Microorganisms can be found in any other part of the environment on Earth, for example in the soil, water, and also the human body. Let's discuss the benefits of microorganisms. Most of the microorganisms are beneficial to man, although some of them could be harmful to man. One of the first benefits of microorganisms is bread production. Microorganisms have been used in the production of bread. In the production of bread, a yeast known as Saccharomyces cerevisiae is used in the production of bread. Then also in the production of alcohol, the same yeast, Saccharomyces cerevisiae, is the one which is used mostly. It's grown from using the fruit sugar. Then in the production of cheese and the yogurt, the lactic acid bacteria has been used for that process, and also streptococcus is used in the initial ripening of cheese. The production of antibiotics and the vaccines. In the production of antibiotics, we have the precaryotes, which are mainly bacteria. They have been used in the production of antibiotics. Then we have the vaccines. Within the vaccines, we have the bacteriotoxoids. These ones have been formed from, for example, Clostridium tetani or Cyanobacteria diphtheriae, which have been used for the production of the Cyanobacteria diphtheria, which is being used as a vaccine against diphtheria. Then the other one has been used as a vaccine against tetanus. Then we have biological control. There are a variety of microorganisms, for example, the agrobacterium, the alkaligens, and Pseudomonas and the Rhizobium. These ones have been used for biological control in that they are used to protect some of the plants against fungal infections. Then the production of gases. Some of the bacteria, for example, the bacterioloids, the Clostridium, some of these bacteria produce gases, and some of these gases are composed of hydrogen, carbon dioxide, and methane. Let's move on to the history of microbiology. In the ancient times, the first recorded observations of microorganisms were made in ancient Egypt and Greece, where people observed the effects of yeast and mold on bread and also wine, especially when they are preparing wine and when the wine gets fermented. In 1665, The Dutch scientist Antonio van Leeuwenhoek used a single lens microscope to observe and describe bacteria and other microorganisms for the first time. Then in 1676, the Italian physician Francisco Redi conducted an experiment that disapproved the theory of simultaneous generation, which held that living organisms could arise simultaneously from non-living matter. Then in 1796, Edward Jenner developed the first vaccine from developed the first vaccine for smallpox, and he developed this vaccine using the cowpox virus as a vaccine. Then in 1657, Louis Pasteur demonstrated that microorganisms were responsible for the fermentation of wine, beer, and he developed the process of pasteurization. And up to today, the process of pasteurization is used to kill harmful microorganisms that are found in liquids. For example, the milk that we drink today has undergone the process of pasteurization to kill the harmful microorganisms. In 1876, Robert Koch developed a series of postulating he developed a series of postulates that are still being used today to prove that particular microorganisms causes specific diseases in humans and also in animals. Let's continue with the history of microbiology. In 1928, Alexander Fleming discovered the first antibiotic, penicillin, which revolutionized the treatment of bacterial infections. The fact after the discovery of penicillin, most of the bacterial infections were able to be treated. Then in 1956, James Watson and Francis Crick, they published their paper describing the structure of DNA, that is deoxyribonucleic acid. This led to the foundation of the field of molecular biology. Let's move on to the classification of microorganisms. Microorganisms can be classified into several groups, and this is based on their characteristics such as the shape, the size, the structure, and function. The main groups of microorganisms include bacteria, viruses, fungi, protozoa, and algae. Classification of microorganisms. Microorganisms have been classified into acellular and cellular organisms. Under the acellular microorganisms, we have examples like the viruses. Then in the acellular and under acellular microorganisms, we have examples like the prebiotic, for example, the bacteria, the archaea. Then under the eukaryotic, we have the examples like algae, protozoa, and fungi. Let's look at the differences and similarities between prekaryotic and eukaryotic cells. Prekaryotic cells, these are small and simple, whereas the eukaryotic cells, these are large and complex. The prekaryotic cells, their size ranges from 0.1 to 5.0 unimicrons in size, whereas the eukaryotic cells, these are larger, they range from 10 to 10 unimicrons in size. The prekaryotic cells, These are unicellular, whereas the eukaryotic cells are unicellular or multicellular. The prekaryotic cells, the nucleus is absent, whereas in the eukaryotic cells, they have a nucleus. The prekaryotic cells have a cellular DNA, whereas the eukaryotic cells have a linear DNA. Then the prekaryotic cells have a single haploid number of chromosomes, whereas the eukaryotic cells have a paired, that is diploid number of chromosomes. The prekaryotic cells lack membrane-bound organelles, whereas the eukaryotic cells have membrane-bound organelles. In other words, the organelles are bound within a membrane. Then the prekaryotic cells reproduce both sexually and asexually, whereas the eukaryotic cells mostly reproduce asexually. Then the prekaryotic cells undergo cell division by the process of binary fusion, whereas the eukaryotic cells undergo cell division by the process of mitosis. Then the examples of prekaryotic cells include bacteria and archaea cells, whereas examples of eukaryotic cells include animal cells and plant cells. The similarities of both prekaryotic and eukaryotic cells are: one, they all have a cell plasma membrane. Also, they have a cytoplasm. They all have ribosomes, and they both have DNA, deoxyribonucleic acid. Let's look at the characteristics of microorganisms. Microorganisms can be spherical, rod-shaped, or spiral-shaped, and they exist as single cells or aggregates. Microorganisms can use various energy sources and metabolic pathways. These include photosynthesis, respiration, and fermentation. All these sources are being utilized, or all these processes are being utilized for the production of energy for the organism, for the microorganisms. Some microorganisms can cause disease in humans and plants, and also in animals. And some microorganisms are harmful and others are also beneficial to the human beings. Characteristics of microorganisms, we are still continuing with that. Remember, microorganisms can evolve very rapidly and they can grow very rapidly. And when they grow or when they evolve, this will lead to new strains. And these strains are usually resistant to antibiotics and they become more virulent than the previous strains. Microorganisms are very tiny living organisms that cannot be seen by the naked eye, but they can only be seen with the help of a microscope. Let's look at bacteria. Bacteria, these are single-celled organisms that can be found in almost any other part on the earth. They can also be found on the human body. Bacteria can be classified based on its shape, staining properties, and other characteristics. But mostly bacteria can be divided into two main groups. We have the gram-positive and gram-negative bacteria. Bacteria, remember we said that bacteria, these are prekaryotic cells, which means that they do not have a nucleus or membrane-bound organelles. Instead, their genetic material is contained within a single cellular chromosome, and this single cellular chromosome is located in the cytoplasm of the bacteria. Let's look at the uses of bacteria. Bacteria can be used to produce antibiotics. Most of the antibiotics that are used for treatment of various infections today have been produced from bacteria, and still more antibiotics are being produced. Streptomyces, these ones have been produced from bacteria and they can be used in the treatment of tuberculosis. Bacteria have been used in the production of enzymes, proteins, vaccines, and these are made through biotechnology. For example, E. coli or Escherichia coli bacteria, these ones have been used to produce human insulin. In agriculture, rhizobium bacteria form a symbiotic relationship with the leguminous plants. Some of the leguminous plants include the beans, the cowpeas, and the rhizobium bacteria fix the atmospheric nitrogen and provide it to the plants as a nutrient. Let's continue with the uses of bacteria. Bioremediation of the soil and water. For example, Pseudomonas, Pseudomonas bacteria, they break the hydrocarbons that occur in oil spills. For example, in the oil drilling areas, once the oil spills into the water, then the Pseudomonas bacteria will break the hydrocarbons that are found in the oil spills, and when they break them down, then it means that they have restored the ecosystem back to its former state. Bacteria are used in production of food and beverages. For example, bacteria are used in the production of yogurt, cheese, and beer, whereby some of the bacteria which are being used is known as the lactobacillus. These ones can be used in the fermentation of milk and also in the production of yogurt and cheese. Let's look at the structure of bacteria. Bacterial cells have several different structures, and these different structures perform specific functions. Some of these bacterial cells include the nucleoid, which contains the DNA, that is the oxyribonucleic acid, the cytoplasm, the ribosomes, the villi, the plasmid, the inclusion borders, flagellum, cytoplasmic membrane, the capsule, and the cell wall. Parts of the bacteria cell. We have the cytoplasm. The cytoplasm is made of a fluid-filled interior of the bacteria cell, and it contains mostly genetic material, ribosomes, various enzymes, and metabolic pathways that are important for the bacterial cell to survive. Then we have the flagella or flagellum when it is one. This is a long wick-like structure that extends from the bacterial cell surface, and it is used for locomotion, and also it helps the bacteria to move towards its nutrients such that it can be able to engulf them or eat them up. and it also helps the bacteria to move away from toxins in case there are toxins within the surrounding of the bacteria, because these toxins can be harmful to the bacteria. That's why it has to quickly move away by the use of the flagella. Let's stop there for now. We'll continue with the parts of the bacterial cell next time. Thank you for listening.